from the Institute of Plant Molecular Biology and Agricultural Biotech here at the Chinese University of Hong Kong. And so about 10 faculties in the science, so we group together work on different aspects of biology, mostly on plant biology. But we focus on rice, obviously, because rice is an important crop. It provides stable food to half the world population, particularly in Asia, where 1/3 of the dietary energy is from rice, and also about 1/4 of the protein is from rice. So we work on various aspects to understand the molecular and cell biology of yield, quality of rice, biotic and abiotic stresses, for example, disease, drought, and salt resistance. But today I will just focus on one of the things I myself is working on, is the quality of rice. So, as you know, rice, about 80% contains starch, and some 7 to 8% is protein. So this is a major protein source for the people who eat rice. And rice protein is inexpensive and it's quite good protein, except that one of the essential amino acid, the lysine, is about 40% below the World Health Organization recommended value. So the limiting essential amino acid in rice protein is lysine. So lysine, a group of amino acids our body cannot synthesize or convert, so we have to intake it from the food. And there are two characteristics of essential amino acid. That is, all the essential amino acids have to present at the same time in our food and also in proper proportion. So then we can make use of these essential amino acids for protein synthesis, etc. But if one amino acid becomes limiting, so proportionally, the rest of the other essential amino acid will be metabolized, cannot be stored or converted to protein. So essential amino acid is critical for our growth and development. If you look at our whole body protein, you can see among the essential amino acid, lysine is pretty high in the human body and some other mammals. So lysine is quite abundant in our whole protein body. And also, we need lysine, different amount at different developmental stage. For example, in children, we need more lysine. And then when we grow up, we need less. But still, we need about 30 milligrams per kilogram of body weight per day. Right? So then, if we are someone with 70 kilograms body weight, so we need about 2 grams of lysine each day, right? If people use rice as the main staple food, to obtain this 2 grams of lysine per day, they have to eat 760 grams of rice per day. That's a lot, because usually for rice eating people, we eat about 200 grams per day. So to get enough, you have to eat quite a bit. So then if we don't have essential lysine, of course, all kind of problem for children's development and growth. Just imagine, right? We cannot make some of the antibodies, proteins, enzymes, so they're bound to be many, many problems. So that is the essential of the lysine. So then if we don't have sufficient proteins and essential amino acids, we may result in malnutrition. And there are some studies showing if we feed the children with this malnutrition with corn, which is high in lysine, then this disease can be alleviated. So lysine indeed is very important. But in rice, we want to really test whether lysine is important in rice for the growth and developments. So that's why we did some feeding experiment with rats. So we provide the diet with rice protein, but we add different amount of free lysine to the rice diet. So here is the table. So the wild type of rice contains such amount of lysine, and then we add 20% equivalent lysine to the rice diet. 20% means the WHO recommend value. So the rice is 20% below, 40% below, so we add 20% back, and the other one 30%, 40%, and a little bit excess. So this will be the diet we try to feed to the animals. And so we do this feeding in such a way we would like to control different group of animals, feeding different amount of lysine supplement to eat the same amount, then we can compare. So in the first 30 days, we try to get the animal to eat the same amount. However, after about a month, it turns out those with add lysine will grow much faster. So they become very hungry. So compare then becomes unfair again because they don't have enough to eat. But anyway, so after 30 days, we give them whatever they want to eat. So the way to try to get them to eat the same amount is we make the diet into powder. So this will be fixed amount so they can eat and finish every day. By that way, we can control what they are eating. So this is after about 60 days feeding. And here is the body weight gain by eating the regular rice. And the other one is adding 20%, 30%, 40%, 50%. So indeed, in about 2 months growth, we can see very significant difference in the growth of the animals. By adding lysine, we can promote their growth, or this is a serious limiting the growth. So we have a problem is the lysine is not enough in rice, and also this study demonstrates if we can add lysine to it, maybe we can enhance the rice nutritional quality. So we have justification appears to undertake this engineering. So we try different ways. So I just want to report 2 of the ways we try to enhance the lysine content in the rice. One is trying to enhance the protein-bound lysine. Means the lysine in the rice is part of the protein. Another way is by increasing the free lysine content in the rice. For this one of the approaches is trying to overexpress protein in rice, which is rich in lysine. So try to raise the level. So then try to increase the level of lysine at the same time. The second one is by metabolic engineering of the lysine synthesis and breakdown pathway, try to control or enhance the synthesis of free lysine. So right now I will try to describe the parts and the technologies we try to do this experiment. So in rice, there are 4 main groups of proteins. So 80% in the rice we are eating is the is the acid or alkali soluble protein. We call glutamine. It's about 80% of the total seed protein. And then the others are water-soluble albumin, salt-soluble globulin, and also alcohol-soluble prolamine. So these are the major storage protein, and there are multifamily proteins in the different groups. And you can see, so the glutamine, this part, they have much more lysine compared to the other fractions. That's why we think this lysine rich, and if we can engineer the rice to make more of this protein, hopefully we will be able to enhance the lysine content in the rice. So this is the glutamine gene. You can see they have about 2.4 mole% of lysine. And so this is the part of the gene we are going to use in this approach. The other one concerning the free lysine, the lysine is synthesized in the plant through the aspartate family amino acids. So the aspartate comes in, and two key enzymes, AK, aspartate kinase, and the DHPase. These two enzymes are the key ones to synthesize the lysine plus some other enzymes. And also, in the rice, there are two enzymes break down the lysine or convert the lysine to other metabolites. So the lysine ketoglutaric reductase and the other SDH. It turns out these AK and DHPase are substrate feedback inhibitor. That means if the rice has more lysine, this is the substrate, this is the product. And then this go back to inhibit the AK and the DHPase. So it turns out, looks like the rice want to maintain certain level of lysine. If it's too high, it goes back to inhibit its synthesis. So this is the characteristic of these two enzymes if we want to engineer. Another one is in rice, the LKR and SDH is a bifunctional enzyme. So it's a protein with both amino acid, both enzymatic activity. So what we're trying to engineer is, so we would like to find enzymes or genes insensitive to the lysine inhibition. So we call this feedback insensitive enzymes. The other one will be, we will use RNAi try to block the function of these. So we will continue to make these and block the degradation of these. By this way, we hope to enhance the free lysine content. So we have studied the developmental expression profile. For example, the AK gene synthesized very high at the mid-maturation stage. Yet the DHPase, the other one, very high during early stage, but then becomes decreasing. So you see the coordination is not very well in the rice. And then the breakdown one is also peaked with the synthesis one. So this gives us insights on how to engineer the coordinate, the expression. And we also try to look at what kind of promoter in the rice we should use. So we look at all kind of rice major proteins expression pathway. So you can see they have different expression profiles. So then we can select those may fit our purpose of expression. With this in mind, so we want to test some of the promoter as a part we would like to use. So we use GUS as a reporter gene and transform plants with different promoters driving the GUS. So then we can cut the rice and look at their specific expression and also their amount. So we pick the glutenin 1 because they express quite strongly and also inside the rice endosperm. So if we engineer any lysine, it should be inside the white, the part we are eating. So we pick promoters according to different things. So then with all these promoters and coding sequence ready, we need to develop transformation procedure for rice. Rice has two major subspecies, Japonica and Indica. This is the shorter grain. The northern China, Japanese use for sushi, and the Indica is the longer grain. So turns out the Chinese hybrid rice, which has 35 to 40% higher yield than regular rice, is all Indica background. So turns out Japonica is much easier to transform, but Indica is very, very hard to transform. So we spend times develop the technology, how to put genes into different subspecies of rice. So at the end, we can transform Japonica very well, very high efficiency. But now we can also transform Indica rice. At least we can obtain enough amounts to use. So with this ready, so we would like to engineer the rice and see whether they will have this lysine enhancement effect. For the expression of a lysine-rich protein, so in the rice, the glutenin will express as a precursor and then process into acidic subunit and also basic subunit. this is the precursor. So this will be what happened in the rice. And so we use vectors to express this gluterin. And for the metabolic engineering of the free lysine, we make different constructs. And with 35S promoter, which is constitutive, expressed in all the tissues all the time. And also we use different promoters according to what we think will be helpful. And we also do the RNAi to block the degradation enzyme. And so we have 3, and then we do combinations. So here will be enhance the synthesis and at the same time block the breakdown with different promoters. And the feedback in sensitive enzyme or genes are from bacteria and provided by Dr. Galili. So then we do the transformation. So now I can show some of the results we have. So in terms of expressing the lysine-rich rice gluterin, we can see individual seeds we harvest. You can see they have, some of the seeds have very high expression of progluterin, the precursor. And then that means the expression is very strong, making a lot of precursor protein. Looks like they cannot process enough into the subunit. But anyway, so this will be the express of the gluterin. And we find out that, so this is the wild type total protein, and these are different transgenic lines. You can see the precursor gluterin expressed a lot, right? And the subunit become less. That's what we observe. And then the turns out, even though the gluterin expressed more, turns out the total protein doesn't change. It turns out the other storage protein was suppressed. You can see the gluterin albumin become less compared to the control, and the globulin also become less, and the prolamine also become less. Maybe it's a good thing because prolamine is very hard to digest this alcohol. So less, turns out, so this compensate by the overexpression of the gluterin. Anyway, when we did the amino acid analysis and compare of the transgenic lines, there are some lines enhance 20% of the lysine, some 27%, and some close to 30%. And the other amino acid doesn't seem to make much big difference. Okay, so this is the protein-bound lysine. For the free amino acids, right, we have different constructs. And we tested in the greenhouse. Turns out, using the 35S promoter with the RNAi give us the best enhancement in the lysine, and then the 6 storage protein promoter and the others are less enhanced. So then different promoters give us different results. So we have to testing out different promoters. So now I would like to show you some of the field trial on this high lysine protein we generate. So we did some small scale, 2 to 3 mole testing in China. Of course, in Hong Kong, we don't have lens for testing. But anyway, so these are the lines we are testing in the field, see whether this enhancement holds up in the real situation. So this is one of the testing in Hainan Island. You can see the free lysine enhanced over 1,000%. And other testing also with significant enhanced, very similar to the greenhouse result. So then lastly, we would like to test animal feeding on this lysine enhanced rice, and turns out certainly they make some difference in the growth. So here is the wild type rice at free lysine that still promote the most, and the others also give significant enhancement. So it turns out the feed efficiency is significantly enhanced by enhancing the lysine content. So the result, again, plot like this. So in the rest few minutes, I would like to show rice, besides they don't have enough lysine, they don't have vitamin A, they don't have enough vitamin E, and have not enough iron and zinc. So for those who only have a bowl of rice to eat, this will cause a lot of problems, visual and other growth problems. So then the Bill Gates Foundation in the Grand Challenging in Global Health, in the Challenging 9, fund 4 projects to enhance the nutritional quality of banana, cassava, rice, and sorghum. All try to enhance the nutritional quality. So for the rice, it's trying to enhance the beta-carotene, the provitamin A, the vitamin E, good protein, enhance iron and zinc. So the idea is to use the golden rice, which contain high beta-carotene already, and we stack in the gene or the tray with vitamin E, iron, zinc, and lysine. So we produce a high-quality protein vitamin rice for the poor people to use. So different countries, different scientists contribute to different part of this engineering, and myself will be contributing those lysine enhancement technology to this project. So I would like to thank my students and also my collaborators and also the funding agency from Hong Kong as well from the Gates Foundation. Thank you very much. Yes.
Sequences to contain a better balance of.
Yeah, I think someone has this idea and tried a long time ago. Syntactic gene they call. So you can put lysine, methionine, essential amino acid. Turns out yes, you can enhance, but that becomes a normal protein. So you have to test a lot, allergy, digestion, right? So it has problem too. Yeah, people try. Of course, right, I can put a string of lysine to it, but then I have to do a lot of things for FDA to approve.